Welcome once again. Good afternoon, good morning, good evening, and happy Halloween to you all. My name is Edgar Napoleon Asimwe. You're welcome to the fifth webinar of HOLICARE Academy and HOLICARE project. We have three speakers today who will be talking about microfluidics. We have Professor Aman Russom, Professor Zulf Ali, and Professor Konstantinos. They will introduce themselves.
My name is Konstantinos Mitsakakis. I'm working as a group leader in immunoassays at the bioanalysis division of Hahn-Schickard and in the laboratory for MEMS applications at IMTech, the Department of Microsystems Engineering at the University of Freiburg in Germany. And the topic of my presentation is the centrifugal microfluidics for global health diagnostics. So just to position the project HOLICARE in this webinar, when we were building the proposal, we tried to handle or to tackle African healthcare challenges by means of the four pillars that also Aman presented in the beginning. So as to demonstrate that diagnostics is really important for those challenges, but it has to be coupled together with other activities such as capacity building, adoption and implementation, and human capital potential. The following slides are focusing on the innovative diagnostics and the pillar A. In particular, the global health and infectious disease diagnosis. So pathogen identification through nucleic acid amplification, either PCR or isothermal, is key for infectious disease evidence-based diagnosis and patient management. For technology developers like us, this means bring the technology into the chip, like lab on a chip, as was mentioned before, and bring it at the point where the care is given. That's the point of care that was also mentioned before. This means that we, or the technology developers, we need to integrate and automate the laboratory operations in order to reduce labor costs, reduce manual errors, minimize the hands-on work and time to result, make the diagnostic tools available to limited trained personnel, and save the time of the personnel to do other crucial tasks while the test is running. So microfluidics, as was mentioned before, can be a driver to such integration. And among the several microfluidic platforms available, centrifugal microfluidics is a good candidate. Why is that? Because they offer a broad palette of physical principles for liquid handling, such as centrifugal forces, capillary forces, pneumatic operations. They're modular and easily adaptable to diverse application needs, and they can be applicable for a wide range of assays, for example, nucleic acid, protein, and cell-based analysis. Typically, how these fluidics are developed is you have a partner, or you yourself, develop an assay, and you have a block diagram like this with the steps of the assay, and then there are specific unit operations, as we call them, microfluidic unit operations, each of them simulating one of these steps. And at the end, all these unit operations are interfaced into a final centrifugal microfluidic disk. And one example of them looks like this, the LabDisk, a fully integrated centrifugal microfluidic platform with all the reagents pre-stored. Because remember, you need to integrate the whole lab into a chip or a disk. So for detecting, let's say, pathogen, you need to lyse them, you need to process the DNA, you need to amplify. So all these reagents need to be included in such a disk. A short video to show how a couple of such operations of the lab disk. Here you will see some liquid mixed with magnetic particles. Then the disk starts to spin. There is some mixing of the bits, and then you can imagine one magnet collecting them, and the second magnet pulling them out, transferring them to the next chamber for the purification of DNA. Then there's a purified liquid, let's say, containing DNA, and then this is by centrifugation aliquoted, metered and aliquoted to reaction chambers, where you can imagine in each one of these you have pre-stored primers, and then let's say with a specific instrument processing the disk, you can also have the real-time amplification on that device. And that device would look like this, roughly. That's what we were calling the LabDisk Player 1, first generation. That was a functional model. It was quite compact, and it consisted of hardware like mechanical unit, of course, for the rotation, thermal unit for the thermocycling or isothermal, detection for fluorescence or chemiluminescence or imaging, as well as built-in magnets for the processing of magnetic particles. So this brief introduction about the LabDisk. Now let's go to the implementation of such a system into the context of global health, which is the title of the presentation anyway. So we will start with the tropical infections and with a scenario. So let's assume that there's a patient who presents with acute fever at resource-limited settings. By default, malaria is assumed and the test is done first. Then let's assume malaria is negative. Then what happens? Would you do an additional test? That would mean additional cost from doing sequential tests. And what if they are not available, those tests? Then you would need to travel to the closest clinic with whatever closest means in such areas. Or maybe the doctor would give any drug that they have in the drawer, just acting under pressure. In addition to that, one could imagine that there are overlapping epidemics of dengue, chikungunya, and so on, which make the diagnostic landscape much more complicated. So acute fever is the same symptom in all these diseases, and the problem is that false diagnosis would lead to wrong drugs, and this would lead to people dying or developing resistances. So that triggered us in this project, Diagnostics, to try to develop a system for the differential diagnosis of acute fever via multiplex identification of pathogen, RNA, and DNA. For this, we chose disease panel, which was consisted of bacteria, parasites, and viruses. So representative were Salmonella, Typhim paratyphim, Streptococcus pneumoniae. From parasites, we selected the Plasmodium, Falciparum, vivax, malaria, responsible for the malaria. And viruses, we had three main ones, dengue, chikungunya, and Zika. So a panel which have all of them have same clinical symptoms, but they require different treatments. So important to be differentiated. That project led to the fever disk. So the design looked like this. Na1 is the inlet with 200 microliters of sample, which was blood or serum. Then lysis of the pathogens and extraction of DNA was happening with the use of buffers, which were stored in packages called stick packs, and this what is named Na2. Na3 shows magnetic particles, which would capture the magnetic beats. And remember the magnets, the built-in magnets mentioned before, they were transferring those magnetic beads along this structure Na4, and eventually the purified DNA RNA was transferred up here, which Na5 is a lyophilized pellet containing all the amplification enzymes. And then this mixture was aliquoted to the area Na6, where 12 chambers were hosting 12 sets of primers for detecting 12 targets, and the detection was done using real-time LAMP isothermal amplification. Eventually, the time to result totally from start to end was 70 to 120 minutes, depending on the assay. We did a small-scale validation in Senegal at the Institut Pasteur in Dakar, who, by the way, is also a partner in HOLICARE. Here you see Ben, one of our project partners, and here is Sheikh, also member of the HOLICARE. We sent 50 disks. The tests were done using collected samples from past epidemics, and the confirmation was done using some reference methods with, depending on the availability, either lateral flow tests or microscopy or benchtop RT-PCR or RT-LAMP. Some indicative outcomes are shown here. Here on the right, you see how we distributed the primer sets along the various amplification chambers. So let's say if we would get, we knew that if we would get a signal coming out of chamber number 9, that would mean that we detect dengue 1. So like here, for example. By the way, we splitted the 4 serotypes of dengue into 4 different chambers. If we would get a signal from chamber 10, that would mean detecting dengue 2 like here. A couple of highlights were, for example, those samples were characterized with RT-PCR before, as I said, because they were biobanked from previous epidemics. So during the prior characterization, one sample was known to be of dengue, but not known of which serotype. So we could manage to serotype it as dengue 1, since we got an amplification from chamber 9. There was a negative chikungunya sample. Here you see 8 chikungunya straight line. But in that sample, we sort of detected or discovered an additional malaria because we got signal from chambers 1 and 2, which were deriving from malaria. And similarly, in a positive chikungunya, we also discovered an additional dengue 2, which had been missed by the initial measurements because, as I said, in an epidemic, you typically screen for the epidemic pathogen. So this is a nice example of why such tools are important to detect co-infections, like in this last case. There was a second validation in Sudan with the help of our project partner MAST, who also are members of HOLICARE. A few discs were sent at the central laboratory in Khartoum for testing malaria primarily. And during the tests, one patient appeared there in real time being sick. He was tested during the measurements using a standard microscopy, was found positive, and then freshly collected sample was used in the disc, and it was confirmed with Plasmodium falciparum with a pretty fast assay time. And lastly, we wanted to demonstrate, let's say, multiple detection of bacteria. So we spiked all the 3 different bacteria at once in 200 microliters whole blood, and we could detect all of them simultaneously with a pretty good limit of detection. So we're talking about malaria, dengue, chikungunya, and Zika, and where do these infections come from? So the answer is from here, from this one, from the mosquito. And there was a new project called DMC Malvec, where we had the motto that humans get sick because mosquitoes transmit. And the problem is that these vector-borne diseases are responsible for more than 17% of all the infectious diseases. And the occurrence of these diseases is not anymore located only in tropical areas, but it moves towards moderate climate zones as well. Of course, the main way to fight them is insecticides, but there are still challenges there. First, there's an increasing development of resistances to insecticides, and second, there is a lack of appropriate mosquito analysis tools. So this is where we were coming in, and we tried with the LabDisk, which in this case was called the vector disk. We tried to help with by providing an automated analytical tool for all necessary levels of molecular-based information for vector management. So in this case, we tried to integrate 28 assays in a disk, which was pretty many. Outside the disk, we performed the smashing of the mosquitoes and the preparation, and then the prepared mosquitoes were inserted into the disk, and then we had again this mixing with lyophilized reagents and the multiplexed detection. In each chamber this time, we had either duplex or triplex RT-PCR compared to the singleplex RT-LAMP before. So what kind of molecular-based information would be needed for an efficient characterization of mosquitoes? First of all, to define if the mosquito is Anopheles gene in case of malaria, which is a DNA assay, or Aedes species if it's an arboviral vector, so an RNA assay. So that was achieved. In case a mosquito was infected with malaria, one would need to determine if the infection was from a Plasmodium falciparum or Vivax or Ovale or malaria, so the species of Plasmodium, which would be a DNA assay, and also the infective stage, how infective the mosquito was, which was an RNA assay, which was also done. And at the end, to assess the insecticide resistance levels, which was done through DNA and RNA assays as well. So eventually, that vector disk was the other side of the same coin of the fever disk, because as we saw, all these vector-borne diseases were also part of the human diagnostic vector disk, fever disk. Then we go to respiratory tract infections as a main candidate of global health threat. Why is that? First of all, the respiratory infections are major causes of hospitalization, either in primary care or in the emergency room. These are the main settings. Main respiratory pathogens are of viral and bacterial nature, and often diagnosis is done symptomatic, but if it's viral or bacterial, it needs a different treatment. So differentiation is needed. Otherwise, antibiotics are often given unnecessarily, like in case of viral infections, which triggers very much the antibiotic resistances. And all of us have heard how this is an increasing trend currently and also in the next years, and Aman mentioned it in the beginning as well. Also, why is a global threat? Because the past, say, 20 years, many of the epidemics that have happened, or pandemics, have been related to respiratory infections. In that context, we expanded the application of the LabDisk towards the RespiDisk. And in this case, we thought, okay, let's try to integrate commercial reagents. What you saw before in the fever disk and vector disk were the agents developed during the project by partners. Now we said, okay, let's see if we can integrate ready-to-use commercial reagents. So we had the panel on the RespiDisk consisting of several viruses and bacteria, corona as well, but not the COVID-19 that was still below or before the COVID era. But we had the other types of and subtypes of corona. For a proof of principle study, we used 36 samples that we bought from QCMD, and they were procured as external quality assessment panels, EQA. We used UTM as a specimen, so the liquid and the reagents, as mentioned, were commercially available. And as reference method, we used the routine clinical diagnostic method of the University Clinic, Uniclinic Freiburg. The RespiDisk looked like this, not very different to the fever disk, some modifications towards optimization using some advanced unit operations. Again, you see sample inlet. This green shows the place where the stick packs were with those buffers. The area 2 was the bind wash elute and purification of nucleic acid. Number 5, the lyo pellet. Number 7, the PCR chambers. In this case, we used an RT-PCR in the RespiDisk. So the main outcomes were that we were testing challenging sample concentrations from the QCMD, which were typically used in commercial devices to test themselves. So the agreement between what we found positive and negative and what QCMD found positive, that's here. What we found positive and negative versus what QCMD found positive and negative was 85%. What Uniclinic found versus what QCMD found was also 85%. And the agreement between our results and the Uniclinic Freiburg was at 100%. So we were pretty fine with those results and published them also in Analyst. Now we've been talking this time about DNA, RNA. Maybe we've been talking too much is the question. Is the DNA and RNA the holy grail really of diagnostics? Remember the phrase we said at the beginning, pathogen identification through nucleic acid is a key for infectious disease evidence-based diagnosis. But the nucleic acid amplification alone may not differentiate colonization versus infection, may not differentiate cases of sepsis, for example, versus non-sepsis inflammations, or may not define disease severity. In these cases, if you have host response biomarkers, which are secreted by the body and not by the external agent, and if you detect them or quantify them in a complementary way with a pathogen ID, then that could improve diagnostic accuracy and efficiency. Great. But let's remember that the pathogen ID, to do it, you need the nucleic acid amplification, and to do a biomarker quantification, you need an immunoassay. So that means you need different instruments, which means costs, and you need different settings to do the analysis. For example, the microbiology for the pathogen ID versus the clinical chemistry to do the biomarker. This means that you receive the results from each of the two at different timeframes, and this may delay the whole process. Eventually, what we were proposing is, what if there is an immunoassay that can be compatible with and integrable with nucleic acid amplification point-of-care instruments? So basically, we have one device which can handle a PCR cartridge and an immunoassay cartridge. To progress with our suggestion, we use CRP as a case study biomarker because it's broadly used for guiding antibiotic prescription. So, for example, cutoff values like are 20, so below 20, typically no prescription. Above 100 milligram per liter, yes. And in between, it's a gray zone where you need to co-assess with other clinical parameters. This is how was born the bound-free phase detection immunoassay, BFPD. And this picture shows the typical competitive format. So you have magnetic particles with coupled antibodies. You have fluorescent particles with coupled competitive antigen. And together with the sample, they are mixed, incubated. And after the incubation, the magnetic beads with whatever is bound on them is removed. And then you measure the supernatant or whatever is unbound fluorescent beads. Therefore, the name bound-free. We tested this with some real samples, clinical samples, quantified using our method, the CIVD ELISA kit and immunoturbidimetric assay commercial analyzer. And we were pretty happy to find a nice correlation between our assay and the immunoturbidimetry, so near one, and also between our assay and the CIVD ELISA kit, also near one. That was on the microtiter plate. Remember, we said a few slides before that it would be important if the same device could process a nucleic acid amplification and immunoassay. So that device could be LabDisk Player 1, for example, proven to process a PCR disk, but what about the host response biomarkers? Well, for those, there is immunodisk, which looks like that. And it's a full disk consisting of 3 structures, identical ones. So in principle, 3 samples on the same disk could be run with fully integrated reagents for sample to answer immunoassay. These are the stored beads, magnetic and fluorescence, and here is a liquid in a stick pack. Typically, the process is you insert your serum in chamber 1, you start spinning the disk. Number 2, the stick pack opens, releases the liquid, the buffer, which comes inwards, mixes with the sample. Then these 2, they go to number 3, they rehydrate the fluorescent beads, and the whole mixture goes to number 4, rehydrates magnetic beads, and then it's the incubation, separation, and detection that we saw before. So it's a pretty simple process. The heart, let's say, is this chamber, which we call it multi-purpose chamber, exactly because it does all these functions. And there's a secondary chamber on top of that, you can see it here maybe, which is a storage chamber, which helps to stabilize those magnetic and fluorescent beads during the drying process. We demonstrated that with CRP with a 20-minute time to answer. We had the standard care where we were below and above the main cutoff values. And we used the certified reference material by the EU with a non-concentration 41.2. With 3 batches, we measured it on average 46.9, which was pretty near. And we had a CV of less than 10%. We were also pretty satisfied with that. So that was an immunodisk on a functional model device. So then the next level of challenge was, how about integrating this assay into a commercial instrument, which was a spin diagronda. That was a device using a half disk for 3 viruses, SARS, RSV, and influenza. And the challenge was to integrate the BFPD on such a device where not really any hardware changes are possible, and also to demonstrate some sort of a throughput because it was a COVID-19 era. So remember before, this was the multi-purpose chamber, the heart of this BFPD assay. And so what if you have less degree of integration, so no stick packs, but you have a higher degree of throughput? So putting 7 such chambers into a half disk, that means 14 data points per run. The reproducibility between all those chambers was measured to be at 5.4%. And we demonstrated a sort of clinical application scenario, the chronic obstructive pulmonary disease, the COPD. And we wanted to semi-quantitatively classify real samples into concentration zones. And these zones were created by 2 cutoff values, the 20 and the 40. And after calculating the LOD at 2.3 milligram per liter, we tested 27 serum samples with a BFPD on the benchtop microtiter plate and on the Ronda. So Among these 27, 22 of the cases were in agreement between the two methods. So the green ones means that they were classified into the right or into the same zone between the concentration zone between the two methods. So as a conclusion, the centrifugal microfluidics, they offer some advantages like modular strategy, multiplexing detection, full automation. They allow minimum hands-on steps, and they are suitable for limited trained personnel. They offer compactness, portability, and point-of-care use. Of course, multi-purpose, because we saw they can be applied in tropical diseases for human and vector diagnosis, in respiratory infections. They can detect DNA, RNA, proteins, different types of pathogens, and different sample matrices: blood, serum, saliva, which I did not mention, swab, and mosquitoes. So eventually, I would like to express my deep acknowledgements to all my colleagues in the past years that have helped very, very much into these developments, the project partners, and of course, the funding agencies.